Hello everyone, and welcome to this podcast for genetics. In this podcast, and the next podcast, we're going to focus on cancer. We're going to talk about its characteristics, we're going to talk about how it forms, and why it can be so devastating. I'm going to take a step back though, and talk a little bit about we had G1, we had S, we had G2, and we had M phase. And in genetics, we primarily focused on meiosis, because that was really important to understand some of the foundations of genetics, and that's still going to be the case. However, as we think about cancer, we're going to want to come back and focus primarily on mitosis, because cancer is a disease of mitosis, or we could say of the mitotic cell cycle. So if you remember, when we talked about the cell cycle, we talked about one cell entering the cell cycle, the DNA copying, and then in mitosis, it would kick out two healthy cells that were genetically identical. We also talked about how, as you're going through this cycle, or maybe due to some sort of environmental factor, that mutations could occur. And as those mutations occur, these cells could become mutated. However, we did talk about how these mutated cells could undergo various repair mechanisms so that instead of being mutated, they were now wild type or not mutated. What we're going to talk about in this podcast is how sometimes these repair mechanisms aren't enough, or sometimes the damage is so great that the repair mechanisms aren't enough. Sometimes they get overloaded. And when that happens, we don't end up with healthy cells. We end up with these mutated cells. And these mutated cells will re-enter this cell cycle, and they will continue to generate cells that have a mutation. And as we talk about cancer in this podcast and the next podcast, we're going to see how these mutations can accumulate. Once you have one, it's much easier to generate new mutations. And as we go forward with this discussion, we're going to talk about very specific genes that when they become mutated, it allows this whole process of forming a cancer to move much faster. Now, before we move forward and continue our discussion about cancer, I'm going to take a moment and watch a short video. This video is going to show you healthy breast cells dividing, and then it's going to show you breast cancer cells dividing. And we're going to look at the differences, and I'm going to stop the video and point out some hallmarks, and then we'll come back to the whiteboard. Okay, let's go ahead and start this video. You'll notice here, these are the healthy cells, and when they divide, they're going to divide at a fairly constant rate. And I'm going to hit pause here. You'll notice that they're forming what appears to be a fairly symmetrical structure. This is what most cells do, and these happen to be cells of the lumen found in mammary tissues. And they're going to show a cartoon of that in just a moment here, of what you would expect it to look like. So here are the cells that line it normally, and these are the cells that we see in this particular video. Now, what they're going to show are cells taken from breast cancer tissue. And you'll see how much faster these cells divide. So let's take a peek at those. As you're watching them, notice the rapid rate at which they're dividing. I'm going to hit pause here. And you can see they don't form a nice, regular, symmetrical structure any longer. So they're dividing faster, and their shape is much different. So let's keep watching this, and then it's going to show it one more time. I'm going to hit pause here, because what we see at the very end here is a very large mass of cells, still very asymmetrical. But you could see how these cells clumping together could form what is characteristic of a lump that a person might feel in their breast tissue as a first sign of cancer. So this is characteristic of what that lump might look like. It's going to show it one more time, and I'm just going to let you watch it without saying anything, but as you're watching it, take special note of how fast it's dividing compared to the healthy cells we saw earlier, and it's at the same time speed, so you can see how fast they are dividing. And then notice how the shape is much different and how it forms this big mass of cells. And then we'll move back to the whiteboard. Okay, that video did a nice job of introducing to you what cancer cells look like, how they divide, how their shape changes compared to healthy normal cells. How what we know about the genetics and the molecular biology of a cell can help explain how a cell can be transformed from a healthy normal cell to a cancerous cell. And as we get started with this, let's talk about just some general facts about cancer. First, let's sort of define cancer here. And that is that cancer is characterized by uncontrolled growth, I should say uncontrolled and rapid growth, of cells. And with that, let's also talk about how cancer cells are morphologically different than other cells, than the cells that they came from. A liver cancer cell looks much different than a liver cell. In fact, they become so different that as cells become cancerous, they will eventually be able to travel to other parts of the body. In fact, it's not really cancer until they do travel and colonize other parts of the body. Up to this point, we've talked about several diseases. We've talked about diseases like xeroderma pigmentosum, Tay-Sachs, cystic fibrosis, and many others. But cancer is different in that cancer is not a single gene disease. If you have a mutation in the Tay-Sachs gene, the child will have Tay-Sachs. Same thing with cystic fibrosis, same thing with sickle cell anemia. However, cancer is not a single gene. You cannot have a mutation in a single gene, and you definitely are going to get cancer. Rather, we know that for cancer to form, we usually require approximately 10 different genes to be mutated. Because of that, we often think of cancer as a disease of old age, because it takes a while to accumulate 10 different mutations. Unfortunately, we all know that kids and even babies sometimes get cancer. So it's not always a disease of old age, but it often is because of the time that it takes to accumulate 10 different mutations in the same cell. Next thing I'd like to say is that cancer involves both genetic and environmental influences. Often the environmental influences will generate mutations, and anytime there's an environmental influence like a chemical of some sort, or UV light, or many of these other agents around the environment, we say that they are carcinogens. Carcinogen is a chemical that generates mutations that can lead to cancer. Okay, next I'm going to erase this so I can make a few other points here, and I'm going to begin on this next whiteboard talking about the inheritance of cancer. Now, in the last slide, we said that there is a genetic influence, that genes get mutated and that can lead to cancer. However, that doesn't necessarily mean that cancer is a hereditary disease. So what I want to say here is cancer is not necessarily a hereditary disease. So what do I mean by cancer not being a hereditary disease? What I mean by this is that most people with cancer didn't inherit a mutated cancer gene, meaning that their parents didn't necessarily have cancer. However, if a person does inherit a cancer-linked gene, their chances of getting cancer increases greatly. So, since most people get cancer without inheriting a mutated allele, that means that throughout the course of their life, they gain those mutations, most likely due to some kind of environmental exposure to some kind of carcinogen. But if you are born with one, that means you are one step closer to developing cancer at some point in your lifetime. Because remember, you need about 10 mutations. So if you're born with one, that just makes it that much easier to get to that critical mass of mutations. The last fact about cancer I'm going to talk about is the incidence of cancer. Now, this varies greatly depending on the kind of cancer. So these are just very general terms. We know that, statistically speaking, about 40% of Americans will get cancer. This number is pretty similar to any industrialized country. We also know that at about 20 to 25% of Americans die from cancer. Cancer is still not the leading cause of death worldwide, but it is a disease that affects a lot of people on this earth. So it's a disease that is important for us to learn more about and try to find ways to stop it or to prevent it. Next, I want to talk about the multi-step model of cancer progression. This was first formulated, this idea, by Knudsen in 1971. Since it's not a single gene mutation, these mutations have to accumulate. And as they accumulate, the cancer cells will take on new traits. So let's begin by looking at some epithelial cells. And I'm just going to draw them looking something like this. So this could be on the skin, the surface of an individual. That'd be a good place to think about it. This could also be epithelial cells that line the intestines or the respiratory tract. But let's just think about it as the skin. We know that each of these cells will divide. And we know that each of them will have a nucleus. So let's go and draw a small little nucleus here. Now, even though cancer is not a single gene mutation, cancer will start off with a single mutation, typically. And this single mutation often is a gene that regulates the cell cycle in one way or another. And we'll come back to that in a little bit. But for now, let's just say that this cell here has a mutation in it. Just one mutation. And maybe that mutation allows it to divide faster than the other cells. So let's write that down over here. The first mutation often affects the cell cycle. And this will allow the cell to divide faster. So remember, all the cells are going to be dividing, but this cell here, and I'm going to draw it in blue just to distinguish it from the others, is going to be dividing faster. And initially, the cell might look quite similar to the normal cells. But now, since it's dividing faster, it has a greater chance to accumulate more mutations. So we'll say more mutations accumulate. And now what happens is that these cells will begin to have an altered appearance because they're taking on new mutations. So I'm just going to draw them so they appear different, not only a different color, but a different shape. Just like when we saw those breast cancer cells. They would look quite a bit different than the normal breast tissue cells. A key step now that happens is that these cells, they're dividing faster. So they're doing two things. They're overcrowding the tissue. So more and more of the cells are now going to be this mutated form. And fewer of them are going to be of the original form. And because of that, they're stealing nutrients from healthy cells. So not only are they growing faster, and that's preventing these other cells from dividing, but they're stealing all the nutrients because they're growing faster. Now, as this tissue is growing, I'm going to draw a neighboring tissue. Now, this neighboring tissue is healthy tissue. It's not cancerous. And what this mutated cell will do now is it will begin to invade even this neighboring tissue. And I'm going to draw the cells even a little more different. I'll keep them green because I only have so many colors. But they will begin to invade these other tissues. And they begin to look really rather abnormal. They will invade other tissues. I'm going to put other neighboring tissues. They will also evade the immune system. But each time they take on a new form or invade a new tissue, it's because they've accumulated more mutations. Now, at some point, what's going to happen is that these cells do something quite clever, but also quite devastating. And is really the reason these cells become cancerous, is that they will begin to secrete chemicals. I'm just going to show that by these these brown dots. They're secreting these chemicals. And what they are chemicals that neighboring blood vessels, and I'll just show this as a blood vessel here, react to. These chemicals will help stimulate a process called angiogenesis. And I'm going to erase this just so I have more room here. So chemicals released stimulate angiogenesis. Angiogenesis is a term used to describe the growth of new blood vessels. This is important when we have tissue that gets injured and we need to build new tissues. So we grow blood vessels to those tissues. It's important during embryonic and fetal development. And the cancer cells hijack that to help recruit blood vessels to these rapidly growing cells. So these chemicals are released, and now the blood vessels will start to be recruited to the rapidly dividing cells. Now, as these blood vessels are being recruited to these rapidly dividing cells, it positively affects the developing tumor and negatively affects the organism. So as these blood vessels are moving up here, they will begin to wrap around these cells. And in doing so, they provide to the dividing cells. They also provide a transport mechanism for the cells. I want to make sure I mention a few terms here that I haven't already. And the first one is a term that we're going to call the primary tumor. The primary tumor is the collection of these rapidly dividing cells within this single tissue. We often call this kind of a tumor benign. It could negatively impact this organ or this set of tissues here, but there's less of a chance that it's going to affect the whole organism. Now, as these blood vessels move here, this allows these rapidly dividing cells to enter the circulatory system. So these cells now are in the circulatory system, and they've accumulated more mutations simply because they've been able to invade a new tissue, the circulatory system. And they're also continuously evading our immune system. Now, once in the circulatory system, they can essentially travel to any part of the body. So maybe over here we have the liver. The liver is a fairly vascularized tissue, and so the blood supply routinely goes through here. And as the blood pumps through the liver, it's also pumping through these tumor cells that have escaped. And some of these cells, not all of them, because it's not easy to do, but some of them will break away from the circulatory system and begin to colonize the liver. And they're going to be growing much faster than the liver cells. And they'll be able to outcompete the normal liver cells here. And at this point, we call this the secondary tumor. We also say that at this point, the cancer has metastasized. Another term for a secondary tumor is called a malignant tumor. And this is cancer. These cells up here are not yet cancer. They're just a benign tumor. But by the time the cancer is metastasized, it is now a malignant tumor, and that is cancer. So in this podcast, at least this part of the podcast where we talked about the multi-step model of cancer progression, we started with a single cell mutation. That single cell mutation started to grow faster than the surrounding cells, started outcompeting the cells for nutrients and space, and then it started invading some neighboring tissues, and finally it started to recruit blood vessels. And these blood vessels allowed these tumor cells to now metastasize and form a malignant tumor elsewhere. Now in the next podcast, we're going to talk about how this cell gains the ability to divide fast. And we're going to talk about some common genes that are found mutated in cancer cells. But that's all I have for this podcast. So if you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.